This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome. Today, we journey deep into the human genome to uncover a remarkable story of survival written in our DNA. We'll explore the MUC19 gene and the mysterious Oreo haplotype, a genetic relay race between Neanderthals, Denisovans, and the first modern humans. Discover how this sticky armor fueled the epic migration across the Americas, and why this ancient legacy remains a powerful yet double-edged sword in modern medicine. Join us as we trace a biological masterpiece that has been tens of thousands of years in the making. Chapter 1: The Oreo haplotype and the multi-species relay race. In the vast landscape of the human genome, researchers have discovered a structure that challenges our traditional understanding of how we inherited our biological traits. This discovery centers on a gene called MUC19, but the story is not found in the gene alone. Instead, it is written in a specific arrangement of DNA known as a haplotype, a cluster of genetic variants that are inherited together like a single, unbreakable package. Scientists have begun to describe this specific arrangement as a genetic Oreo. In this molecular sandwich, a core of 72,000 base pairs of Denisovan-specific code is nested inside a much larger 742,000 base pair region of Neanderthal DNA. This nested architecture is the first documented evidence of a multi-species genetic relay race, revealing that some of our most important survival tools were passed through multiple extinct relatives before they ever reached the modern human lineage. To appreciate the precision of this discovery, we must look at the specific smoking guns hidden within the code. Within the 72 kilobase Denisovan cream at the center of the Oreo, researchers identified exactly 9 non-synonymous variants. These are mutations that do not merely sit silently in the background. They are diagnostic signatures that change the actual protein produced by the gene. These 9 specific changes are unique to the Denisovans, a mysterious sister group to the Neanderthals known primarily from DNA extracted from bone fragments in Siberia. Finding these 9 Denisovan markers embedded within a massive 742 kilobase block of Neanderthal code provides an unmistakable map of ancient encounters. It tells us that the genetic material did not come to us directly from a Denisovan, nor did it come from a standard Neanderthal. It came from a hybrid lineage that had already merged these two ancient codes. This Oreo structure provides a definitive timeline for a sequence of events that occurred tens of thousands of years ago. Typically, we think of interbreeding as a simple exchange between two groups, a human meeting a Neanderthal or a human meeting a Denisovan. However, MUC19 reveals a more complex inheritance. The evidence for this relay race is found by comparing different ancient genomes. When scientists examined the high-coverage genome of the Altai Neanderthal, an individual who lived in the Denisova cave roughly 120,000 years ago, they found that it did not carry this Denisovan-like variant. However, when they looked at later Neanderthals, specifically those from the Chagirskaya cave in Siberia and the Vindija cave in Croatia, they found that these individuals were heterozygous for the Oreo haplotype. They carried one copy of the standard Neanderthal version and one copy of the hybrid Denisovan Neanderthal version. This distinction is crucial. It proves that the interbreeding event between Denisovans and Neanderthals happened in a specific window of time, and likely in a specific geographic region. Because the Altai Neanderthal lacked the variant while the Chagirskaya and Vindija Neanderthals possessed it, we can see the relay in action. The Denisovan code was introduced into a specific branch of the Neanderthal population, which then carried it forward for generations. By the time modern humans migrated out of Africa and encountered Neanderthals in Eurasia, this Oreo package was already a stable part of the Neanderthal toolkit, ready to be passed into the human line. Understanding the difference between a gene and a haplotype is essential to grasping why this discovery has fascinated the scientific community. While MUC19 is the gene, the specific sequence that provides instructions for building a protein, the Oreo is the haplotype, the broader context in which that gene sits. In the human genome, recombination usually acts like a deck of cards being shuffled, breaking up long segments of DNA over time. The fact that this massive 742 kilobase Neanderthal segment containing the 72 kilobase Denisovan core has remained intact is a significant biological signal. When a segment of DNA this large survives the shuffling process and reaches high frequencies in a population, it suggests that the entire package was being actively preserved by natural selection. The MUC19 gene itself is a massive and complex piece of biological machinery. It belongs to the family of gel-forming mucins, which are the primary components of the protective barriers in our bodies. These proteins are heavily covered in complex sugar molecules, allowing them to form the thick, resilient substance we know as mucus. This substance acts as a frontline defense, protecting internal tissues from pathogens and irritants. Because MUC19 is a prime candidate for evolutionary pressure, an ancient group that developed a version of this gene better suited to a specific environment would have a powerful advantage. The discovery of the Oreo haplotype shows that modern humans did not have to wait for the slow, random process of mutation to develop these advantages. In the standard model of evolution, a beneficial trait can take tens of thousands of years to appear and spread. But interbreeding, or adaptive introgression, serves as a genetic shortcut. By inheriting the Oreo haplotype, early humans borrowed a biological toolkit that had already been refined and tested by Denisovans and Neanderthals over hundreds of thousands of years. This multi-species hand-me-down provided a sudden infusion of genetic variation that was already adapted to the challenges of the Eurasian landscape. This research highlights the power of modern computational biology. By using new tools to analyze high-quality ancient DNA, scientists can now see patterns that were invisible just a few years ago. We are no longer just looking for Neanderthal DNA or Denisovan DNA in general terms. We are identifying specific, nested structures that reveal the braided nature of our ancestry. The MUC19 Oreo is the first of its kind, a documented case where we can see the exact scale of the genetic package, from the 742 kb cookie to the 72 kb cream and the 9 specific mutations that make it work. The presence of this variant in modern humans is not uniform. While it is rare in many parts of the world, its geographic distribution suggests that while the gene was forged in the ancient encounters of Eurasia, its true value was realized much later, as humans moved into entirely new environments. The Oreo haplotype wasn't just a neutral passenger in the human genome. It was a piece of ancient technology that became indispensable under specific environmental pressures. As we examine this multi-species inheritance, we are forced to rethink the nature of our extinct relatives. The Denisovans and Neanderthals were not merely primitive versions of ourselves who vanished into the past. They were biological innovators whose genetic contributions continue to function in the bodies of millions of people today. The MUC19 Oreo is a tangible link to those ancient cousins, a record of a time when the boundaries between human groups were porous and genetic traits were traded like tools. The story of this gene is a testament to the resilience of the human lineage and our ability to absorb the strengths of those who came before us. We are a mosaic of these ancient streams of DNA, and the Oreo haplotype is one of the most sophisticated pieces of that mosaic ever discovered. It represents a successful relay race across species and across millennia, carrying a biological legacy from a Siberian cave into the modern world. But even as we map the structure of this genetic sandwich and trace its path through the Neanderthal line, a deeper question remains. If this gene was so beneficial that it was preserved across 3 different species and maintained in such a large, unbroken block of DNA, what exactly was the pressure that made it so essential? What was it about the role of MUC19 as a protective barrier that allowed this specific Denisovan Neanderthal hybrid to outcompete the standard human version? To understand the true power of this Oreo gift, we have to look beyond the code and follow the people who carried it as they moved toward the edge of the known world. What challenges did they face that forced the human genome to lean so heavily on this ancient inheritance? Chapter 2: The Biological Gauntlet, a Legacy Forged in Ice. The crossing of the Bering Strait is often described as one of the most harrowing chapters in the human story. Approximately 25,000 years ago, a small group of pioneers moved from the familiar landscapes of East Asia into the vast icy bridge of Beringia. For thousands of years, they lived in a state of isolation, hemmed in by massive glaciers, waiting for a path to open into the unknown. When the ice finally retreated, these ancestors didn't just walk into a new continent, they stepped into an entirely different world. Every plant they gathered, every animal they tracked, and every microscopic pathogen they inhaled was a mystery. They were the first humans to ever set foot in North and South America, and their survival depended on more than just stone tools and fire. It depended on a biological resilience that had been forged over hundreds of thousands of years. We now know that as they descended from the glaciers and fanned out across the Americas, they carried a specific genetic legacy in the MUC19 gene. To understand the magnitude of this migration, we have to recognize that the Americas represent the ultimate environmental gauntlet. Within a few thousand years, these early populations moved from the dry, subarctic Beringian steppe through the temperate forests and massive grasslands of the interior, eventually reaching the humid tropical rainforests of the Amazon and the high-altitude peaks of the Andes. No other human migration in history required such rapid adaptation to so many different biomes. While cultural ingenuity, like the development of new hunting technologies or agricultural techniques, was vital, recent scientific research suggests that their bodies were also adapting at a deep molecular level. The MUC19 gene appears to have been a central player in this survival story. The significance of this find cannot be overstated. In the world of evolutionary biology, finding a gene that has been so clearly favored by natural selection is rare. The statistical signals of this selection are among the strongest in the human genome, showing that the environment was actively choosing this gene over the standard version. While the technical proof of this selection is found in the modern genomes of millions of people today, the story begins with the Beringian standstill. This hypothesis suggests that the ancestors of indigenous Americans were isolated for up to 10,000 years before they entered the continent. During this time, the population was small and the environment was brutal. MUC19 didn't just survive this standstill, it was refined by it. By the time the path to the south opened, the population was already equipped with a genetic package that had already been tested in harsh environments. This allowed them to hit the ground running. Instead of waiting for thousands of years for new mutations to slowly appear, they had a biology that was ready for the challenges ahead. As these pioneers moved south, they weren't just experimenting with new types of food. They were encountering entirely different ecological niches that demanded a versatile biological defense. A gene that could provide a more robust protective barrier would have been a massive advantage, whether a person was breathing the thin air of the Andes or the humid air of the Amazon. Those who carried this specific version were likely better equipped to handle the respiratory and gastrointestinal challenges of the New World, making them more likely to survive and pass their genes on to the next generation. The fact that the MUC19 variant persisted and even increased in frequency as people moved through these radically different biomes suggests that its benefits were universal. It was a biological insurance policy against the unknown. This brings us to a fascinating realization about the nature of human expansion. The colonization of the Americas wasn't just a journey across a landscape. It was a journey through a deep reservoir of genetic variation that had been accumulating for hundreds of thousands of years. We often frame the success of the first Americans as a triumph of the human spirit, of bravery and ingenuity. And while that is true, the MUC19 research adds a new layer of depth to that narrative. It shows that our survival is also a triumph of our biology's ability to utilize the best possible tools. We are a species that doesn't just adapt to our surroundings, we absorb the strengths of those who came before us. This continuity is one of the most complex stories in all of science, connecting ancient pioneers in Beringia to modern people in the Americas today. But to truly understand the power of this gene, we have to look closer at what happened to the protein itself once it reached the Americas. While the ancient foundations provided the starting point, the gene underwent a further massive transformation that is unique to the American journey. Why would the body need to take an already powerful gene and expand its functional power even further? What was it about the American biomes that demanded such an extreme biological response, leading to a molecular change that exists nowhere else in the world? Chapter 3: The Architecture of Sticky Armor. To understand the biological potency of the MUC19 gene, one must look past the history of its inheritance and examine the physical blueprint of the protein itself. While the ancestry of the gene provides the context, its internal architecture provides the function. Recent structural analysis has revealed that MUC19 is a molecular giant, possessing a structural complexity that begins at the very start of the protein chain. Human MUC19 possesses an unusually long amino terminus located upstream of the first von Willebrand factor D (VWD) domain. This specific structural feature is entirely absent in other mucins like MUC2 or MUC5B. This extended front end makes MUC19 uniquely elongated even among its peers, providing a massive scaffold for biological interactions before the protein even reaches its primary functional domains. The true engine of MUC19's power, however, lies in its central repetitive region. This area is defined by variable number tandem repeats, or VNTRs, genetic stutters where a specific sequence of DNA is repeated dozens or even hundreds of times. In MUC19, the fundamental unit of this repetition is a 30 base pair sequence. In the language of the cell, every 3 base pairs translate into 1 amino acid, meaning each repeat adds a 10 amino acid link to the growing protein chain. What makes the variant found in the Americas so extraordinary is the scale of this expansion. While the archaic genetic background provided the template, a massive expansion of these repeats occurred on that specific haplotype. This expansion effectively doubled the length of the protein's functional domain, transforming a standard protein structure into a reinforced fortress. This repetitive domain is not just a filler, it is a dense forest of biochemical hooks. The 10 amino acid units are exceptionally rich in 2 specific residues, serine and threonine. These amino acids are the essential docking sites for O-glycosylation, the process by which the body attaches complex sugar molecules to a protein core. When these sugars are added, the protein core is transformed into its functional state. Because the American variant has doubled the number of these repeats, it has doubled the number of serine and threonine hooks available. This results in a protein that is more heavily armored with sugars than almost any other protein in the human body. The resulting glycoprotein is larger, more resilient, and capable of forming a much denser molecular mesh. The sheer scale of this stutter is best illustrated by comparing the human gene to its counterparts in other species. In the mouse version of the gene, known as MUC19, each repeat encodes a staggering 163 residues. While the human repeats are shorter in individual length, the human gene has evolved to include a far greater number of them, creating a much longer overall transcript. The human MUC19 gene is a behemoth, consisting of 182 exons and spanning a transcript of approximately 25,000 base pairs. At its heart is a massive central exon, a single uninterrupted stretch of code 12,000 base pairs long. In most genes, exons are short segments separated by non-coding introns, but MUC19 maintains this giant unbroken block to ensure the repetitive domain remains perfectly continuous. Any interruption in this sequence would break the rhythm of the hooks, compromising the structural integrity of the resulting gel. The versatility of MUC19 is further enhanced by a process known as alternative splicing, where the body edits the gene's 182 exons to create different versions of the protein for different tasks. Scientists have identified 20 different splicing variants in the highly variable region of the gene. This allows the body to deploy a diverse toolkit of protein shapes from a single genetic source. Perhaps the most fascinating example of this versatility is the connection between MUC19 and a salivary protein known as SMGC. Research has shown that MUC19 and SMGC are actually splice variants of the same larger gene complex. This means the same genetic machinery responsible for the massive sticky armor in the lungs is also responsible for producing specialized proteins in the salivary glands. This shared origin highlights MUC19 as a master of mucosal management, capable of shifting its form to meet the specific demands of different tissues. This structural doubling has profound physical consequences for the rheology of the resulting substance, the way it flows and deforms. The density of the sugar-coated VNTRs determines the pore size of the molecular mesh. A standard version of MUC19 creates a mesh that can trap larger particles and some bacteria. However, the expanded variant, with its doubled functional domain, creates a much tighter, stickier net. This sticky armor is significantly more effective at binding to pathogens and neutralizing them before they can reach the underlying cell membranes. For a population encountering a new environment, this wasn't just a minor adjustment, it was a fundamental upgrade to the body's frontline defenses. This strategy of using internal tandem repeats to create biological glue is a proven evolutionary tactic seen elsewhere in the natural world. A striking parallel is found in the stickleback fish, which produces a specialized nesting glue called spigin. Like MUC19, the spigin protein is derived from a mucin-like gene that evolved through duplication and expansion. While the fish uses this glue to build physical structures in its environment, humans use MUC19 to build a microscopic structure within their own bodies. The human gene, however, opted for internal expansion, duplicating the repeats within the gene rather than duplicating the entire gene itself. This is a more efficient way to increase the protein's power without the metabolic cost of managing multiple gene copies across the genome. The physical work of this expanded MUC19 is most visible in the mouth and the respiratory tract, though the mechanics in each are distinct. In the salivary glands, MUC19 is a primary component of the bioactive fluid that manages the oral microbiome. Saliva must be viscous enough to lubricate the mouth during speech and swallowing, but it must also be sticky enough to aggregate bacteria. The expanded MUC19 variant is exceptionally good at this aggregation. It binds to common oral microbes, preventing them from forming biofilms on the teeth and gums. By doubling the functional domain, the body gains a superior ability to manage the internal garden of the mouth, a vital advantage when a population begins consuming entirely new types of flora and fauna. In the lungs, the mechanics of MUC19 are even more critical. The airways are lined with a mucociliary escalator, a system where a layer of mucus traps inhaled irritants and is then moved upward by the rhythmic beating of tiny hairs called cilia. The effectiveness of this escalator depends entirely on the physical properties of the mucus. If the gel is too watery, it cannot trap pathogens. If it is too stiff, the cilia cannot move it. The expanded MUC19 variant appears to optimize this balance. Because it is more heavily glycosylated, it can retain more water while maintaining a high degree of elasticity. This creates a gel that is tough enough to trap fine dust and microscopic spores, but slick enough to be cleared efficiently from the deep recesses of the lungs. The statistical association between the number of these VNTRs and the archaic haplotype suggests that this expansion was not a random event. Instead, the expansion occurred specifically on the background of the Denisovan-like gene. This suggests that the archaic version of the gene provided a stable structural template that was particularly receptive to these internal duplications. Once the expansion took hold, the environmental pressures of the Western Hemisphere acted as a filter, favoring those individuals who carried the doubled version of the protein. The result was a rapid molecular transformation that reached its pinnacle in the ancestors of indigenous Americans. The existence of this massive, uninterrupted central exon also speaks to the purity of the protein's function. In most of the genome, long stretches of DNA eventually accumulate junk sequences or mutations that break up the code. The fact that 12,000 base pairs of MUC19 have remained a single, continuous coding block indicates that its repetitive nature is essential. The body requires that long, unbroken chain of serine and threonine hooks to build a glycoprotein of a specific length and strength. Any break in the chain would result in a shorter, weaker protein that could not form the necessary sticky armor. This structural requirement is likely why the gene was so receptive to VNTR expansion. The environment simply pushed the repeat button on a template that was already designed for length. Beyond acting as a physical trap, the expanded structure of MUC19 also influences how the body's immune system perceives threats. Mucins are known to interact with receptors on the surface of immune cells, acting as signaling molecules that can prime the body for a faster response. With twice the number of functional units, the expanded variant may be twice as effective at alerting the immune system to the presence of an invader. This dual role, acting as both a physical trap and a biological alarm, makes the MUC19 expansion one of the most sophisticated defensive upgrades in the human lineage. As we look at the intricate, repetitive structure of this giant protein, we see a record of molecular resilience. We see a gene that has been elongated, reinforced, and doubled in power to meet the challenges of a new world. It is a specialized biological response that exists nowhere else in the human family in such an extreme form. But while this sticky armor provided a clear advantage in the wild, its prevalence in modern populations raises a new set of questions. If this gene was so heavily favored by evolution that it became a dominant feature of the American genetic landscape, how does it behave today? Does this ancient doubling of power still provide a benefit in the modern world? Or has the environment changed so much that our sticky armor is now reacting to threats that no longer exist? Chapter 4. The American Map. A Denisovan Legacy Rediscovered. When we look at the genetic map of the world, we expect to see certain patterns. We expect to find Neanderthal DNA in nearly everyone outside of Africa, and we expect to find Denisovan DNA, primarily in populations across East Asia and Oceania, particularly in places like Papua New Guinea. But the story of MUC19 has revealed a massive, hidden legacy in a place where it was least expected, the Americas. Recent scientific research has uncovered a startling reality. A specific, ancient version of the MUC19 gene is present at incredibly high frequencies in modern indigenous and admixed American populations. This isn't just a trace amount or a genetic fluke. In some regions, this ancient Denisovan gift is so prevalent that it has become a defining feature of the local genetic landscape, outshining almost any other archaic signal found in the human genome.
The question immediately became, why here? Why would a gene variant from an extinct group of humans who lived in Asia tens of thousands of years ago be 30 times more common in Mexico than in Europe? The researchers knew they had to dig deeper into the concept of ancestry. Modern populations in the Americas are often admixed, meaning their genomes are a mosaic of indigenous American, European, and African heritage. By using a technique known as ancestry painting, scientists were able to visualize the different segments of these modern genomes to see where the MUC19 gene was sitting. The result was definitive. The Denisovan-like variant was almost exclusively found on the segments of DNA that were inherited from indigenous American ancestors. It wasn't coming from the European or African streams of the gene pool. It was a pure, ancient legacy of the first people who settled the continent. This discovery is particularly striking because for a long time the Americas were considered a bit of a dead zone for Denisovan research. The MUC19 research has completely changed that narrative. It suggests that the Americas might actually be one of the best places on Earth to study how ancient interbreeding shaped modern humans. Earlier studies identified this specific region of the genome as possessing one of the highest densities of Denisovan alleles found anywhere in the Mexican genome. The unique history of the continent, a small group of founders entering a vast land, created a perfect natural experiment for evolution. In this environment, any gene that provided a survival advantage would be boosted to high frequencies by natural selection much faster than it would in the crowded, interconnected populations of Eurasia. This discovery is particularly striking because for a long time the Americas were considered a bit of a dead zone for Denisovan research. The MUC19 research has completely changed that narrative. It suggests that the Americas might actually be one of the best places on Earth to study how ancient interbreeding shaped modern humans. Earlier studies identified this specific region of the genome as possessing one of the highest densities of Denisovan alleles found anywhere in the Mexican genome. The unique history of the continent, a small group of founders entering a vast land, created a perfect natural experiment for evolution. In this environment, any gene that provided a survival advantage would be boosted to high frequencies by natural selection much faster than it would in the crowded, interconnected populations of Eurasia. The recovery of this ancient DNA involves meticulous physical work. Maria Avila Arcos, a lead researcher in the field, has spent years inspecting pre-Hispanic remains, including ancient jawbones recovered from archaeological sites across Mexico and Peru. This labor, extracting fragile strands of DNA from centuries-old bone, is what allowed scientists to bridge the gap between the living and the dead. These ancient individuals lived hundreds, and in some cases thousands, of years before the arrival of Europeans or Africans. The results from these remains were even more dramatic than the modern data, showing the Denisovan-derived MUC19 variant at its highest recorded frequency. This research also highlights a historical irony in the field of science. For decades, indigenous and admixed populations in the Americas were significantly understudied in genomic research. Most of the early breakthroughs in understanding Neanderthal and Denisovan DNA came from studying European and East Asian genomes. By overlooking the Americas, we were missing one of the most important chapters of our own story. The MUC19 gene was hiding in plain sight in the genomes of millions of people. It took a dedicated effort to look at underrepresented populations to realize that the Denisovans had left a much larger footprint on the world than we ever imagined.
This work is part of a broader movement to ensure that the benefits of genomic research are shared by all populations and that we have a truly global understanding of human evolution. The prevalence of this gene also provides a new way of looking at the Denisovans themselves. Because we have so few fossils of this group, mostly just teeth and bone fragments, we have to reconstruct them through their DNA. By seeing how their version of MUC19 functions in modern humans, we are essentially looking at a living piece of Denisovan biology. We are learning about their body chemistry, their immune defenses, and how they interacted with their environment. The fact that their version of MUC19 was so successful in the Americas tells us that the Denisovans had developed a very powerful and versatile set of biological tools. They weren't just a primitive cousin. They were a highly adapted species whose genetic innovations were so robust that they could help modern humans colonize a new world tens of thousands of years after the Denisovans themselves had vanished. As scientists look at the distribution of MUC19 across the Americas today, they are also seeing the impact of recent history. In populations with higher levels of European or African admixture, the frequency of the Denisovan variant tends to be lower. This is because the influx of new genetic material over the last 500 years has created an admixture dilution effect, thinning the ancient signal. However, the fact that it still remains at 33% in Mexico and 20% in Peru is a testament to just how deeply embedded this gene was in the pre-contact population. It was so widespread that even after centuries of massive demographic change, it remains a dominant feature of the genome. It is a biological anchor that connects modern people to the very first pioneers who explored the continent. But why did this legacy survive so well in Mexico and Peru, but not as much in Colombia or Puerto Rico? The research suggests this is tied to the specific demographic history of these regions. In Mexico and the Andean highlands of Peru, indigenous populations were very large and settled, and they maintained a high degree of genetic continuity even after the arrival of Europeans. In contrast, populations in the Caribbean and parts of South America experienced more dramatic shifts and higher levels of admixture, which led to the ancient MUC19 signal being more heavily diluted. This geographic variation is a reminder that the hidden legacy of the Denisovans is not a monolith. It is a complex, shifting map that reflects the unique history of every region and every community. The study of MUC19 also forced researchers to refine their methods for detecting ancient DNA. For a long time, it was difficult to distinguish between DNA that came from a Neanderthal and DNA that came from a Denisovan because the two groups were so closely related. But the identification of Denisovan-specific variants embedded within a larger archaic region allowed scientists to trace the exact lineage of this gene. This has provided a new gold standard for how to study complex multi-species introgression. It shows that we can't just look for ancient DNA in general. We have to look for the specific sequences and patterns that tell the story of how these genes were traded and refined across species. This hidden legacy also has profound implications for how we define modern humans. If 30% of a modern population carries a functional protein-coding gene from a Denisovan, can we really say that the Denisovans are extinct? In a very real biological sense, they are still here. Their innovations are still protecting human lungs and mouths. Their response to ancient environments is still shaping how modern humans respond to the world. The MUC19 gene is a bridge across time, a reminder that the boundaries between species are much more porous than we once thought. We are not just the descendants of the humans who left Africa. We are the sum of all the different human groups who met, shared their lives, and traded their genes along the way. The research into the prevalence of MUC19 is also opening up new doors in personalized medicine. Because this gene is involved in immune defense and the production of mucosal barriers, knowing whether an individual carries the expanded Denisovan variant or the standard human variant could be important for understanding their health. By recognizing that this gene is a major component of the genetic makeup of indigenous and Latino populations, doctors and researchers can begin to tailor their approach to health in a way that respects and understands this unique evolutionary history.
As we look at this hidden legacy, we also have to consider the resilience of the indigenous populations who carried it. For 25,000 years, these groups adapted to every biome on the planet, from the high-altitude deserts to the tropical coastlines. They did this with a combination of cultural brilliance and biological flexibility. The MUC19 gene was a partner in that journey. It survived the expansion into the Americas and the massive upheavals of the last 5 centuries. It is more than just a piece of DNA. It is a record of a population that refused to be erased. But even as we celebrate the survival of this ancient gift, a new and more complex chapter is beginning to emerge. We have spent this narration looking at how MUC19 helped our ancestors survive and thrive in a wild, untamed world. We have seen how it doubled its power to create sticky armor, and how it became a dominant feature of the American genome. But the same qualities that make a gene a powerful tool for survival in the wilderness can sometimes make it a liability in the modern world. As scientists move their focus from the ancient past to the clinical present, they are finding that the MUC19 gene is appearing in some unexpected places, not just in our immune system, but in the pathology of modern diseases. How did a gene for survival become a signal for disease, and what does that tell us about the future of our evolutionary journey? Chapter 5: The Double-Edged Sword of Ancient Armor.
In the grand theater of evolution, every biological advantage comes with a cost. This is the concept of the double-edged sword. A robust immune response that once fought off prehistoric plagues may manifest today as a debilitating autoimmune disorder. As we look at the legacy of the MUC19 gene, from its hybrid origins to its massive expansion in the first pioneers of the Americas, we are forced to confront this evolutionary trade-off. The very biological shield that allowed ancestors to conquer a new world is now emerging in the modern clinic as a critical biomarker for some of our most aggressive diseases. To understand how a survival advantage transforms into a clinical liability, we must look at the sheer scale of the MUC19 gene. It is a genomic giant. In the context of modern cell biology, this massive size makes the gene a significant target for mutations. Because it is so long and repetitive, there are more opportunities for errors during cell division. When these errors occur in a gene central to cell signaling and tissue protection, the results can be catastrophic. One of the most striking examples of this is found in a rare and highly aggressive malignancy known as hepatoid adenocarcinoma of the stomach, HAS. This is a peculiar cancer where stomach cells take on the characteristics of liver cells, producing alpha-fetoprotein, AFP, a marker normally associated with fetal development or liver tumors. Recent genomic profiling revealed that MUC19 is one of the top 5 most frequently mutated genes in HAS, appearing in a staggering 40% of cases. Crucially, these mutations are almost undetectable in standard stomach adenocarcinoma, colorectal cancer, and hepatocellular carcinoma. This makes MUC19 not just a participant in the disease, but a specific diagnostic beacon for this rare subtype. The relationship between MUC19 and this aggressive cancer is functional. Researchers discovered that mutations in MUC19 are positively correlated with high levels of AFP and actually drive the cancer's malignancy. When MUC19 is mutated or overexpressed in these cells, it triggers the WNT beta-catenin pathway, a master switch for cell growth and movement. When MUC19 flips that switch, it tells the cancer cells to grow faster and invade surrounding tissues. In this context, the adhesive nature of the protein may actually help cancer cells adhere to new sites in the body, facilitating metastasis. The stakes for these patients are incredibly high. Hepatoid adenocarcinoma of the stomach carries a 5-year survival rate of only 9%. The reach of MUC19 in oncology extends beyond the digestive tract. In breast cancer, researchers have identified a complex molecular interaction where a circular RNA, HSACIRCOO7534, acts as a sponge for MIR593. By soaking up this microRNA, the circular RNA promotes the expression of MUC19, which in turn fuels the progression of the cancer. This adds a third major cancer type to the MUC19 portfolio, reinforcing its status as a modern clinical biomarker. However, the story of MUC19 in the clinic is not entirely one of tragedy. In a twist of biological irony, the same mutations that drive aggression can also make a tumor more vulnerable to modern medicine. This is most clearly seen in non-small cell lung cancer. For years, doctors have struggled to predict which patients will respond to immunotherapy, specifically anti-PD-1 inhibitors. These drugs unmask the tumor, allowing the immune system to recognize and destroy it. Recent studies have shown that patients whose lung tumors harbor mutations in the MUC19 gene have a significantly better response to this treatment. Specifically, these patients show a hazard ratio of 0.3 for progression-free survival, meaning they are 70% less likely to see their disease progress compared to those without the mutation. The reason for this goes back to the gene's ancient role in immunity. When MUC19 is mutated in a lung tumor, it appears to act as a beacon, leading to an increased infiltration of CD8-positive T cells, the assassin cells of the immune system, into the tumor. The mutated MUC19 protein, with its massive repetitive structure, likely becomes a neoantigen, a foreign-looking target that the immune system cannot ignore. Beyond cancer, MUC19 is a critical player in chronic inflammatory and autoimmune diseases. In patients with Sjogren's syndrome, where the immune system attacks moisture-producing glands, MUC19 is a key factor. The gene is responsible for maintaining the protective film on the surface of the eye. When its production is disrupted, it leads to the severe dryness and tissue damage characteristic of the disease. The same complex regulation that once allowed the gene to adapt to different biomes now makes it vulnerable to the runaway inflammation of an autoimmune response. We see a similar pattern in the respiratory tract. While the robust defense of MUC19 helped ancient pioneers survive new pathogens, the overproduction of this barrier in the modern world can lead to chronic illness. In children suffering from recurrent middle ear infections or otitis media, MUC19 expression is significantly elevated. When the body is flooded with inflammatory signals like TNF-alpha and IL-1 beta, the MUC19 gene goes into overdrive. It produces a thick, viscous gel that natural clearing mechanisms cannot handle, leading to glue ear, which can cause hearing loss and developmental delays. The ancient defense mechanism is still working, but in a modern environment of close quarters and constant respiratory irritants, that defense becomes a source of distress. The impact of MUC19 also extends to common allergies and asthma. While much of the research focuses on the Americas, a significant study in individuals of Russian ethnicity linked specific polymorphic variants of MUC19 to a predisposition for asthma. Similarly, patients with allergic rhinitis show elevated levels of MUC19 in their nasal epithelial cells. It appears that the gene's high variability, which was an asset during the colonization of new continents, now contributes to the wide range of ways people respond to modern allergens. Some variants produce a barrier that is perhaps too sensitive, leading to the airway inflammation that plagues millions today. Even the digestive system remains under its influence. Research has identified a signal on chromosome 12 centered on the MUC19 gene associated with an increased risk for Crohn's disease. This is a reminder that the mucosal barrier is an integrated system. A genetic variant that changes the physical properties of mucus in the mouth or lungs can have far-reaching consequences for how the gut interacts with its resident bacteria. The discovery of MUC19's role in these conditions is changing the definition of a biomarker. MUC19 teaches us that biomarkers can be as complex as the evolutionary history they represent. To understand a patient's risk, we must look at the whole package, the length of their tandem repeats, and the specific ancient variants they inherited. However, the complex history of human migration adds a layer of complexity. Because modern populations are a mosaic of different ancestries, the prevalence and therefore the predictive power of MUC19 as a biomarker varies across different individuals and groups. As we look to the future, the study of MUC19 is moving into the laboratory of functional biology. Scientists are now planning to test these ancient variants in living cells to see exactly how the Denisovan mutations change the properties of human mucus. They want to know if the expanded variant changes the way we fight off modern viruses like influenza, or if the doubled domain makes the protein more likely to trigger an inflammatory response. These answers will bridge the gap between ancient history and the modern hospital ward. This journey through the code of MUC19 has shown that we are a living archive of every migration and survival struggle our ancestors ever faced. We carry the biological legacy of pioneers who crossed into a world of mystery. And we carry the double-edged sword of an evolution that is never finished with us. We are still profoundly shaped by a few thousand base pairs of DNA traded between species tens of thousands of years ago. The past is never truly past. It is active in our spit, in our lungs, and in our struggle against disease. As we stand at the threshold of this new era of evolution-informed medicine, we have to ask, what other ancient gifts are waiting to be discovered in the understudied corners of the human map? If one gene like MUC19 can tell a story this vast, spanning 3 species, 2 hemispheres, and the entire history of human health, how many more sequences are out there, hiding in plain sight, waiting to tell us who we really are and where we are going?
The story of MUC19 is a testament to the braided nature of human history. From its origins as a multi-species Oreo haplotype to its expansion in the first pioneers of the Americas, this gene reveals how we borrowed ancient technology to survive. By doubling its protective power, MUC19 became a sticky armor that defines the biological resilience of millions today. Yet this ancient gift is a double-edged sword, influencing both our immune defenses and our susceptibility to modern diseases. It reminds us that we are not a single isolated lineage, but a living mosaic of every relative we met along the way. As we continue to map these hidden legacies, we must ask, what other ancient secrets are still working within us, silently shaping our future? Thank you for listening.